Hello students, this is Professor Johnson, and in this video we are going to be talking about photosynthesis and some of the historical experiments that have led to our current understanding of how photosynthesis works. So by the end of this video, you should be able to describe photosynthesis overall in a balanced reaction. You should be able to describe the experiments that we're going to talk about and explain what conclusions we could draw from these experiments. We're also then going to look at the light-dependent and independent reactions, which are going to take place in the chloroplast, and you should be able to identify where they are taking place within the chloroplast. So photosynthesis can be summarized in this equation. So it's very much the opposite of cellular respiration. We will take 6 carbon dioxides and 6 waters and end up producing 6 oxygen and glucose in the presence of light. So in cellular respiration, we took oxygen, glucose, we broke those apart into carbon dioxide and water, and that released the energy that we then used to make ATP. That was an exergonic reaction because it released enough energy that ATP could be produced. So if we reverse the equation into photosynthesis now, we should realize that photosynthesis is an endergonic reaction. And that's why light is necessary. We need to put energy into the system to overcome the lack of energy that is going to be produced from these reactions. We need to provide energy for the reactions to actually occur. All right, the first experiment we're going to talk about was performed by an individual known as John Baptista van Helmont. He was interested in knowing where the mass of a plant comes from. So if you take a seed, you plant it in soil, after several years, that seed is going to grow into a tree if it's a tree seed. He wanted to know where all that mass came from. So he performed this experiment where he took a pot and added soil into it. He weighed the soil before. He planted a seed into it. He covered the pot and would only open up the cover in order to allow water to come into the soil. So the only thing he added soil. After several years, he found that the seed had grown into a full tree. So he would then weigh the tree and he weighed the soil as well. The tree had gained several kilograms of mass, whereas the soil had only lost a very minuscule amount, grams or less. So he concluded, because the only thing that he had added was water, that the mass of the tree must be coming from water. We now know that this is an incomplete picture of it because he didn't take account something that was in the air. So Joseph Priestley was looking at gases and how they could affect living organisms. So he found that if he put a mouse in a sealed container with a candle, lit the candle, the mouse would end up suffocating and dying, and the candle would also become unlit. So the mouse would die, the candle wouldn't be able to be lit. So he concluded that the candle and the mouse were both using something up in the air that was vital for their continued sustenance, for the candle, for the flame to keep burning, and for the mouse for it to keep living. However, when he did this with a plant, he didn't find the same results. The plant was able to survive, and actually it was able to keep the candle burning. Furthermore, if he would have put a mouse in here, the mouse would have been able to live as well. So the plant seemed to be producing whatever the mouse in the candle was using. We know that that is now oxygen. So the mouse was using oxygen, the plant is putting the oxygen out. All right, so this is the experiment by F.F. Blackman. He was looking at how carbon dioxide and light intensity could affect the rate of photosynthesis. If we look at the x-axis, we can see that it is light intensity, and the y-axis is the rate of photosynthesis. He looked at several conditions. So he looked at a low carbon dioxide condition, that's the blue line here, a moderate carbon dioxide condition, that's the orange line, and a high carbon dioxide condition, that's the gray line. There's also this yellow line here that shows excess carbon dioxide. So after a point, adding more carbon dioxide doesn't increase the rate of photosynthesis. So he would increase the light intensity, and as he did that, he found that the rate of photosynthesis came up. Now, at certain points, the graphs here would start to separate themselves. So if you had low carbon dioxide, it would level out here at about 20% of what the maximum rate of photosynthesis could be. If he had a little bit more carbon dioxide, it took longer before it leveled out and the rate wouldn't increase anymore. And then if you had as much carbon dioxide as the plant needed, it would eventually level out as well. So what we can take away from this experiment are essentially two things. One is that there are two parts to photosynthesis. So you can see that there's the part over here that depends on light. So it increases with light intensity. Over here, we see that it does not increase with light intensity. However, we can still get the rate of photosynthesis to increase, and that's by adding carbon dioxide. So this tells us that there are light-dependent reactions and reactions that are functioning independent of light. Now, we also know that the light-independent reactions depend on carbon dioxide, because if we give the plant more carbon dioxide, the rate of photosynthesis will increase. We know that the light-independent reactions don't depend on carbon dioxide, because if we look, and this is best illustrated looking at the moderate CO2 and the high CO2 conditions, adding more carbon dioxide here where light was the limiting factor didn't separate the rate of photosynthesis. So something's being produced during the light-dependent reactions that ultimately get used by the light-independent reactions, which depend on carbon dioxide. All right, C.B. Van Niel was looking at a process that was similar to photosynthesis that occurred in purple sulfur bacteria. So up here on the top equation, we have the traditional photosynthesis equation. You'll notice that I've simplified this a little bit so that we're just looking at water and carbon dioxide producing oxygen and glucose or carbohydrate. So I haven't put the C6H12O6. I balanced this out just to make it a little bit more simplified. The process that Van Niel was looking at took, instead of water, dihydrogen sulfide and carbon dioxide. It ended up producing two sulfurs instead of oxygen. You'd still get the glucose, but then you'd also get water here on the product side. Both of these reactions depended on the presence of light. Now, Van Niel made a leap and said that he thought that photosynthesis up here was actually functioning very similar to how this other process in the purple sulfur bacteria was functioning. And so he came up with this generic equation where the H2S here is replaced with H2A, and you have two of those. The carbon dioxide still being used. You produce two of these A substances, and then your glucose and water. And so how this translates to the photosynthetic process is that we have two waters over here on the reactant side. We have carbon dioxide. We produce oxygen. We also produce the carbohydrate glucose, and we produce water. So what ends up happening, and Van Niel was right in his assumption here, is that the water is being used and produced. So the net reaction says that we are using more water than we're producing, and that's why there's only one water up here on this first equation, but we're actually using the water and producing a water because the molecules are being shifted around. So the water that's being ends up here is from different atoms than the water that started over here as the reactants. All right, this is the experiment done by Robin Hill that is now known as the Hill equation. Robin Hill was looking at the light-dependent and light-independent reactions and what they actually depended on. And so he came up with this idea of isolating chloroplast and exposing them to light in the absence of carbon dioxide. When he did this, he found that there was something being oxidized, an electron acceptor, and something that's oxidized, the electron acceptor, that could be reduced into something else. So the electron acceptor gets reduced in this process, and this process would also produce oxygen. How he tested this was he took a dye known as DCPIP that could be reduced. So it is blue in its oxidized form, it's green in its reduced form. If he exposed the chloroplast to this dye, instead of reducing what they normally do, they end up reducing this DCPIP, and you see the color change indicating that something was definitely being reduced. So if we look at what this actually is, and there's a little typo on this image I must apologize for, is that so this is still carbon dioxide being absent. The light exposed chloroplast, we're taking ADP, adenosine diphosphate, and an inorganic phosphate. They're taking NAD, and this should be P+. They were then using those and forming ATP and then the NADPH. So he ended up reducing this NAD plus to NADPH. No sugar is being produced. He is producing ATP, and he's also producing oxygen. If he took these chloroplasts out of the light and put them in the dark now, added CO2 into the mixture, the ATP and NADPH would then be used by the chloroplast. So we get the ADP back, and the NADP plus would be back as well. The CO2 would get formed into the glucose molecule, so we're going to produce sugar, and so photosynthesis could occur. All right, so what does this mean for the overall reaction of photosynthesis? So we divided it up into the light-dependent and light-independent reactions. The light-dependent reactions could be summarized as taking water, NADP plus, and ADP, and producing oxygen, NADPH, and ATP. So NADPH is an electron carrier. It has the electrons. ATP is an energy storage molecule. That is ultimately what is being produced that the plant is going to use for the rest of photosynthesis. The oxygen here can be thought of as a byproduct. So this NADPH and ATP get used by the light independent reaction. So NADPH, ATP, and now we're going to add carbon dioxide into this mixture. We're going to use up the NADPH, so we take the electrons from it, form NADP+. We take the energy from ATP and form ADP, and we use those to form the glucose molecule. And so now we can see if you take out the NADPH and ATP from both sides of the equation, NADP plus and ADP from both sides, we have the water and the carbon dioxide forming oxygen and glucose. So where is this occurring? Of course, it's occurring in the chloroplast. So the chloroplast is a multi-membraned organelle. If we look at the structure of the chloroplast, the granula structures here, so that's what these stack of discs are called, they are formed from the thylakoid membrane. So the thylakoid membrane is where the light-dependent reactions occur. The stroma here outside of the thylakoid is then where the light-independent reactions occur. All right, I hope that was informative. In the next video, we will be looking at in more detail what is going on during the light-independent and light-dependent reactions. I will see you guys in class.